Hello everyone. In this segment we're going to be covering the cellular level of organization. It's chapter 3 from the Principles of Anatomy and Physiology, 12th edition. I've said it time and time again. I'm not showing the illustrations, but in this book they're really kind of good. They're great. So without further ado, this is meant to be audio, so you can listen to it in your car. There's no production value. It is straight audio for now, and maybe later on we'll change it, but I think it's more valuable as an audio tool for you to use in your vehicle, in your home, while you sleep, whatever, an extra study guide. So here we go. The cellular level of organization, chapter 3. Cells in homeostasis. About 200 different types of specialized cells carry out a multitude of functions that help each system contribute to the homeostasis of the entire body. At the same time, all cells share key structures and functions that support their intense activity. In the previous chapter, you learned about the atoms and molecules that comprise the alphabet of the language of the human body. These are combined into about 200 different types of words called cells, living structural and functional units enclosed by a membrane. All cells arise from existing cells by the process of cell division, in which one cell divides into two identical cells. Different types of cells fulfill unique roles that support homeostasis and contribute to the many functions and capabilities of the human organism. Cell biology is the study of cellular structure and function. As you study the various parts of a cell and their relationship to one another, you will learn that cell structure and function are intimately related. In this chapter, you will learn that cells carry out a dazzling array of chemical reactions to create and maintain life processes, in part by isolating specific types of chemical reactions within specialized cellular structures. As I turn the page, I don't think it's covered in this book, it might be, but mutualism, symbiosis, I think you would call it, is one theory that's involved in the creation of cells. It's interesting in that it's theorized that at one point the cell existed on its own, the human cell. An organism, a separate organism, found its way into the cell, into the membrane, as a way of finding nutrients. It found that it could live there, and it never had to leave. All the nutrients came to it, all its waste was carried away, and the cell benefited from it. And it's theorized that that organism later became the mitochondria, and that possibly occurred with many other organisms as well. So that is an interesting theory that further supports that humans are a concert of organisms that live together. We are one person, we are one unit, but we are many creatures that make up the one unit. Now let's carry on. Parts of the cell. Name and describe the main parts of a cell. Figure 3.1 provides an overview of the typical structures found in the body's cell. Most cells have many of the structures shown in the diagram, but no one cell has all of them. For ease of study, we divide the cell into three main parts: plasma membrane, cytoplasm, and nucleus. The plasma membrane forms the cell's flexible outer surface, separating the cell's internal environment inside the cell from the external environment outside the cell. It is a selective barrier that regulates the flow of materials into and out of the cell. This selectivity helps establish and maintain the appropriate environment for normal cellular activities. The plasma membrane also plays a key role in communication among cells and between cells and their external environment. The cytoplasm. The cytoplasm consists of all the cellular contents between the plasma membrane and the nucleus. This compartment has two components, systole and organelles. Systole, the fluid portion of cytoplasm, contains water, dissolved solutes, and suspended particles. Surrounded by systole are several different types of organelles. Each type of organelle has a characteristic shape and a specific function. Examples include the cytoskeleton, ribosomes, endoplasmic reticulum, Golgi complex, lysosomes, peroxisomes, and mitochondria. The nucleus is a large organelle that houses most of a cell's DNA. Within the nucleus, each chromosome, a single molecule of DNA associated with several proteins, contains thousands of hereditary units called genes that control most aspects of cellular structure and function. We're going to move on to the plasma membrane next, but before we do, I'd like to read off all of the components of the cell. The cytoskeleton, microtubules, microfilament, intermediate filament, microvilli, centrosome, pericentriolar material, centrioles, plasma membrane, secretory vesicle, lysosome, smooth endoplasmic reticulum, peroxisome, mitochondrium, microtubule, microfilament, Golgi complex, ribosome, rough endoplasmic reticulum, cytoplasm, systole plus organelle except the nucleus, glycogen granules, nucleus, nuclear envelope, nuclear pore, chromatin, proteasome, cilium, and flagellum. The plasma membrane. The objects of this system, the objective, is to describe the structure and functions of the plasma membrane, explain the concept of selective permeability, define the electrochemical gradient, and describe its components. The plasma membrane, a flexible yet sturdy barrier that surrounds and contains the cytoplasm of a cell, is best described by using a structural model called the fluid mosaic model. According to this model, the molecular arrangement of the plasma membrane resembles an ever-moving sea of fluid lipids that contain a mosaic of many different proteins. Some proteins float freely like icebergs in the lipid sea, whereas others are anchored at specific locations like boats at a dock. The membrane lipids allow passages of several types of lipid-soluble molecules, but act as a barrier to the entry or exit of charged or polar substances. Some of the proteins in the plasma membrane allow movement of polar molecules and ions into and out of the cell. Other proteins can act as signal receptors or adhesion molecules. The structure of the plasma membrane. The lipid bilayer. The basic structural framework of the plasma membrane is the lipid bilayer, two back-to-back layers made up of three types of lipid molecules: phospholipids, cholesterol, and glycolipids. About 75% of the membrane lipids are phospholipids, lipids that contain phosphorus. Present in smaller amounts are cholesterol, about 20%, a steroid with an attachment, hydroxyl group, and various glycolipids, about 5%. Lipids with attached carbohydrate group. The bilayer arrangement occurs because the lipids are amphipathic. Amphipathic molecules, which mean that they have both polar and nonpolar parts. In phospholipids, the polar part is the phosphate containing head, which is hydrophilic, meaning water-loving. The nonpolar parts are the two long fatty acid tails, which are hydrophobic. Phobic equals fearing. Hydrocarbon chains. let me read that again. The nonpolar parts are the two long, fatty tails, which are hydrophobic hydrocarbon chains. Because, like seeks like, the phospholipid molecules orient themselves in the bilayer with their hydrophilic heads facing outwards. In this way, the heads face a watery fluid on either side, systole on the inside and extracellular fluid on the outside. The hydrophobic fatty acid tails in each half of the bilayer point towards one another, forming a nonpolar hydrophobic region in the membrane's anterior. The fluid mosaic arrangement of lipids and proteins in the plasma membrane. Membranes are fluid structures because the lipids and many of the proteins are free to rotate and move sideways in their own half of the bilayer. That layer consists of the lipid bilayer that we spoke of earlier, channel protein, pores, extracellular fluid, glycoprotein carbohydrate protein, peripheral protein, glycolipid carbohydrate lipid, phospholipids, polar heads, which are hydrophilic, fatty acid tails, which are hydrophobic, and polar heads, which are hydrophilic. There's also cholesterol, peripheral protein, integral proteins, and systole. Cholesterol molecules are weakly amphipathic and are interspersed among other lipids in both layers of the membrane. The tiny OH group is the only polar region of cholesterol, and it forms hydrogen bonds with the polar heads of phospholipids and glycolipids. The stiff steroid ring and hydrocarbon tail of cholesterol are nonpolar. They fit among the fatty acid tails of the phospholipids and glycolipids. The carbohydrate groups of glycolipids form a polar head. Their fatty acid tails are nonpolar. Glycolipids appear only in the membrane layer that faces the extracellular fluid, which is one reason the two sides of the bilayer are asymmetric or different. Arrangement of the membrane proteins. Membrane proteins are classified as integral or peripheral according to whether they are firmly embedded in the membrane. Integral proteins extend into or through the lipid bilayer among fatty acid tails and are firmly embedded in it. Most integral proteins are transmembrane proteins, which mean they span the entire lipid bilayer and protrude into both a systole and extracellular fluid. A few integral proteins are tightly attached to one side of the bilayer by covalent bonding to the fatty acids. Let me read that again. A few integral proteins are tightly attached to one side of the bilayer by covalent bonding to fatty acids. Like membrane lipids, integral membrane proteins are amphipathic. Their hydrophilic regions protrude into either the watery extracellular fluid or the systole, and their hydrophobic regions extend among the fatty acid tails. As their name implies, peripheral proteins are not as firmly embedded into the membrane. They associate more loosely with the polar heads of membrane lipids or with integral proteins at the inner or outer surface of the membrane. Many membrane proteins are glycoproteins, proteins with carbohydrate groups attached to the ends that protrude into the extracellular fluid. The carbohydrates are oligosaccharides. Oleo meaning few, saccharides meaning sugars. The carbohydrates are oligosaccharide chains of 2 to 60 monosaccharides that may be straight or branched. The carbohydrate proteins of glycolipids and glycoproteins form an extensive sugary coat called the glycocalyx. The The pattern of carbohydrates in the glycocalyx varies from one cell to another. Therefore, the glycocalyx acts like a molecular signature that enables cells to recognize one another. For example, a white blood cell's ability to detect a foreign glycocalyx is one basis of the immune response that helps us destroy invading organisms. In addition, the glycocalyx enables cells to adhere to one another in some tissue and protects cells from being digested by enzymes in the extracellular fluid. The hydrophilic properties of the glycocalyx attract a film of fluid to the surface of many cells. This action makes red blood cells slippery as they flow through the narrow blood vessels and protects cells that line the airways and gastrointestinal tract from drying out. Functions of membrane proteins. Generally, the types of lipids in cellular membranes vary only slightly. In contrast, the membranes of different cells and various intracellular organelles have remarkably different assortments of proteins that determine many of the membrane functions. Some integral membrane proteins form ion channels, pores or holes through which specific ions such as potassium ions can flow to get into or out of the cell. Most ion channels are selective. They allow only a single type of ion to pass through. Other integral proteins act as carriers, selectively moving a polar substance or ion from one side of the membrane to the other. Carriers are also known as transporters. Integral proteins, called receptors, serve as cellular recognition sites. Each type of receptor recognizes and binds to a specific type of molecule. For instance, insulin receptors bind the hormone insulin. A specific molecule that binds to a receptor is called a ligand, or that receptor. A specific molecule that binds to a receptor is called a ligand of that receptor. Some integral proteins are enzymes that catalyze specific chemical reactions at the inside or outside surface of the cell. Integral proteins may also serve as linkers, which anchor proteins in the plasma membrane of neighboring cells to one another or to protein filament inside and outside the cell. Peripheral proteins also serve as enzymes and linkers. Membrane glycoproteins and glycolipids often serve as cellular identity markers. They may enable a cell to recognize other cells of the same kind during tissue formation or to recognize and respond to potentially dangerous foreign cells. The ABO blood type markers are one example of cellular identity markers. When you receive a blood transfusion, the blood type must be compatible with your own. In addition, peripheral proteins help support the plasma membrane, anchor integral proteins, and participate in mechanical activities such as moving materials and organelle within the cell, changing cell shape, and dividing in muscle cells and attaching cells to one another. I'm going to repeat that. In addition, peripheral proteins help support the plasma membrane, anchor integral proteins, and participate in mechanical activities such as moving materials and organelle within cells, changing cell shape and dividing in muscle cells and attaching cells to one another. Membrane fluidity. Membranes are fluid structures. That is, most of the membrane lipids and many of the membrane proteins easily rotate and move sideways in their own half of the bilayer. Neighboring lipid molecules exchange places about 10 million times per second and may wander completely around a cell in only a few minutes. Membrane fluidity depends both on the number of double bonds in the fatty acid tail of the lipids that make up the bilayer and on the amount of cholesterol present. Each double bond puts a kink in the fatty acid tail, which increases membrane fluidity by preventing lipid molecules from packing tightly into the membrane. Membrane fluidity is an excellent compromise for the cell. A rigid membrane would lack mobility, and a completely fluid membrane would lack the structural organization and mechanical support required by the cell. Membrane fluidity allows interactions to occur within the plasma membrane, such as the assembly of membrane proteins. It also enables the movement of the membrane components responsible for cellular processes such as cell movement, growth division, and secretion, and the formation of cellular junctions. Fluidity allows the lipid bilayer to self-seal if torn or punctured. When a needle is pushed through a plasma membrane and pulled out, the puncture site seals spontaneously and the cell does not burst. When a needle is pushed through a plasma membrane and pulled out, the puncture site seals spontaneously and the cell does not burst. The property of the lipid bilayer allows a procedure called intracytoplasmic sperm injection to help infertile couples conceive a child. Scientists can fertilize an oocyte by injecting a sperm through a tiny syringe. It also permits removal and replacement of a cell's nucleus in cloning experiments, such as ones that created Dolly, the famous cloned sheep. Despite the great mobility of membrane lipids and proteins in their own half of the bilayer, they seldom flip-flop from one half of the bilayer to the other, because it is difficult for hydrophilic parts of membrane molecules to pass through the hydrophobic core of the membrane. This difficulty contributes to the asymmetry of the membrane bilayer. I'm going to read that again. Despite the great mobility of the membrane lipids and proteins in their own half of the bilayer, they seldom flip-flop from one half of the bilayer to the other because it is difficult for hydrophilic parts of membrane molecules to pass through the hydrophobic core of the membrane. This difficulty contributes to the asymmetry of the membrane bilayer. Because of the way it forms hydrogen bonds with neighboring phospholipids and glycolipid heads and fills the space between bent fatty acid tails, cholesterol makes the lipid bilayer stronger but less fluid at normal body temperature. At low temperatures, cholesterol has the opposite effect. It increases membrane fluidity. Membrane permeability. The term permeable means that a structure permits the passage of substances through it, while impermeable means that a structure does not permit the passage of substances through it. The permeability of the plasma membrane to different substances varies. Plasma membranes permit some substances to pass more readily than others. This property of membranes is termed selective permeability. The lipid bilayer portion of the membrane is permeable to nonpolar, unchanged molecules such as oxygen, carbon dioxide, and steroids, but is impermeable to ions in large, unchained polar molecules such as glucose. That would be uncharged polar molecules such as glucose. It is also slightly permeable to small, uncharged polar molecules such as water and urea, a waste product from the breakdown of amino acids. This slight permeability to water and urea is an unexpected property since they are polar molecules. These two small molecules are thought to pass through the lipid bilayer in the following way. As the fatty acid tails of the membrane phospholipids and glycolipids randomly move about, small gaps briefly appear in the hydrophobic environment of the membrane's interior. Water and urea molecules are small enough to move from one gap to another until they have crossed the membrane. Transmembrane proteins that act as channels and carriers increase the plasma membrane's permeability to a variety of ions and uncharged polar molecules that, unlike water and urea molecules, cannot cross the lipid bilayer unassisted. Channels and carriers are very selective. Each one helps a specific molecule or ion to cross the membrane. Macromolecules such as proteins are so large that they are unable to pass across the plasma membrane except by endocytosis and exocytosis, which will be discussed later in the chapter. Gradients across the plasma membrane. The selective permeability of the plasma membrane allows a living cell to maintain different concentrations of certain substances on either side of the plasma membrane. A concentration gradient is a difference in the concentrations of a chemical from one place to another, such as from the inside to the outside of the plasma membrane. Many ions and molecules are more concentrated in either the systole or the extracellular fluid. For instance, oxygen molecules and sodium ions are more concentrated in the extracellular fluid than in the systole. The opposite is true of carbon dioxide molecules and potassium ions. The plasma membrane also creates a difference in the distribution of positively and negatively charged ions between the two sides of the plasma membrane. Typically, the inner surface of the plasma membrane is more negatively charged and the outer surface is more positively charged. A difference in electrical charge between two regions constitutes an electrical gradient. Because it occurs across the plasma membrane, this charge difference is termed the membrane potential. As you will see shortly, the concentration gradient and electrical gradient are important because they help move substances across the plasma membrane. In many cases, a substance will move across a plasma membrane down its concentration gradient. This is to say, a substance will move downhill from where it is more concentrated to where it is less concentrated to reach equilibrium. Similarly, a positively charged substance will tend to move towards a negatively charged area, and a negatively charged substance will tend to move towards a positively charged area. The combined influence of the concentration gradient and the electrical gradient on movement of a particular ion is referred to as its electrical chemical gradient. Transport across the plasma membrane. The objective of this section is to describe the processes that transport substances across the plasma membrane. Transport of materials across the plasma membrane is essential to the life of a cell. Certain substances move into the cell to support metabolic reactions. Other substances that have been produced by the cell for export or as cellular waste products must move out of the cell. Substances generally move across cellular membranes via transport processes that can be classified as passive or active depending on whether they require cellular energy. In passive processes, a substance moves down its concentration or electrical gradient to cross the membrane using only its own kinetic energy, the energy of motion. Kinetic energy is intrinsic to the particles that are moving. There is no input of energy from the cell. An example is simple diffusion. In active processes, cellular energy is used to drive the substances uphill against its concentration or electrical gradient. The cellular energy used is usually in the form of ATP, adenosine triphosphate. An example is active transport. Another way that some substances may enter and leave cells is an active process in which tiny spherical membrane sacs, referred to as vesicles, are used. Examples include endocytosis, in which vesicles detach from the plasma membrane while bringing materials into the cell, and exocytosis, the merging of vesicles with the plasma membrane to release material from the cell. Passive processes, the principle of diffusion. Learning why materials diffuse across membranes requires an understanding of how diffusion occurs in a solution. Diffusion is a passive process in which the random mixing of particles in a solution occurs because of the particles' kinetic energy. Both the solutes, dissolved substances, and the solvent, the liquid that does the dissolving, undergo diffusion. In a particular solute, if a particular solute is present in high concentrations in one area of a solution and in low concentrations in another area, solute molecules will diffuse towards the area of lower concentration. They move down the concentration gradient. After some time, the particles become evenly distributed throughout the solution, and the solution is said to be at equilibrium. The particles continue to move about randomly due to their kinetic energy, but their concentrations do not change. For example, when you place a crystal of dye in a water-filled container, the color is most intense in the area close to the dye because its concentration is higher there. At increasing distances, the color is lighter and darker because the dye concentration is lower. Sometimes a solution of water and dye will have a uniform color because the dye molecules and water molecules have diffused down their concentration gradients until they are evenly mixed in solution. They are at equilibrium. In this simple example, no membrane was involved. Substances may also diffuse through a membrane if the membrane is permeable to them. Several factors influence the diffusion rate of substances across the plasma membrane. Steepness of the concentration gradient. The greater the difference in concentration between the two sides of the membrane, the higher the rate of diffusion. When charged particles are diffusing, the steepness of the electrochemical gradient determines the diffusion rate across the membrane. Principles of diffusion. At the beginning of our experiment, a crystal of dye is placed in a cylinder of water. It dissolves and then diffuses from the region of high dye concentration to the region of low dye concentration. At equilibrium, the dye concentration is uniform throughout random movement. At equilibrium, the dye concentration is uniform throughout, although random movement continues. We've all seen that by putting food coloring in a glass of water. It's the same. Temperature. The higher the temperature, the faster the rate of diffusion. All of the body's diffusion processes occur more rapidly in a person with a fever. Mass of the diffusing substance. The larger the mass of the diffusing particles, the slower its diffusion rate. Smaller molecules diffuse more rapidly than larger ones. Surface area. The larger the membrane surfaces area available for diffusion, the faster the diffusion rate. To repeat that, the surface area. The larger the membrane surface area available for diffusion, the faster the diffusion rate. For example, the air sacs of the lungs have a larger surface area available for diffusion of oxygen from the air into the blood. Some lung diseases, such as emphysema, reduce the surface area. This slows the rate of oxygen diffusion and makes breathing more difficult. Diffusion distance is another variable. The greater the distance over which diffusion must occur, the longer it takes. Diffusion across a plasma membrane takes only a fraction of a second because the membrane is so thin. In pneumonia, fluid collects in the lungs. The additional fluid increases the diffusion distance because oxygen must move through both the built-up fluid and the membrane to reach the bloodstream. Now that you have a basic understanding of the nature of diffusion, we would consider three types of diffusion: simple diffusion, facilitated diffusion, and osmosis. Simple diffusion. Simple diffusion is a passive process in which substances move freely through the lipid bilayer of the plasma membrane of cells without the help of membrane transport proteins. Nonpolar hydrophobic molecules move across the lipid bilayer through the process of simple diffusion. Some molecules, oxygen, carbon dioxide, and nitrogen gases, fatty acids, steroids, and fat-soluble vitamins, A, D, E, and K, once again, in simple diffusion, nonpolar hydrophobic molecules move across the lipid bilayer through the process of simple diffusion. Such molecules I apologize, sorry. Simple diffusion. Nonpolar hydrophobic molecules move across the lipid bilayer through the process of simple diffusion. Such molecules include oxygen, carbon dioxide, nitrogen gases, fatty acids, steroids, and fat-soluble vitamins, A, D, E, and K. Some uncharged polar molecules such as water, urea, and small alcohols also pass through the lipid bilayer by simple diffusion. Simple diffusion through the lipid bilayer is important in the movement of oxygen and carbon dioxide between blood and body cells, and between blood and air within the lung during breathing. It also is the route for absorption of some nutrients and excretion of some wastes by body cells. Facilitated diffusion. Solutes that are too polar or highly charged to move through the lipid bilayer by simple diffusion can cross the plasma membrane by a passive process called facilitated diffusion. In this process, an integral membrane protein assists a specific substance across the membrane. The integral membrane proteins can either be a membrane channel or a carrier. Channel-mediated facilitated diffusion. In channel-mediated facilitated diffusion, a solute moves down its concentration gradient across the lipid bilayer through a membrane channel. Most membrane channels are ion channels, integral transmembrane proteins that allow passage of small inorganic ions that are too hydrophilic to penetrate the nonpolar interior of the lipid bilayer. Each ion can diffuse across the membrane only at certain sites. Its typical plasma membrane, the most numerous ion channels, are selective for potassium chloride, potassium or chloride. In typical plasma membranes, the most numerous ion channels are selective for potassium or chloride ions. Fewer channels are available for sodium ions or calcium. Diffusion of ions through channels is generally slower than free diffusion through the lipid bilayer, because channels occupy a smaller fraction of the membrane's total surface area than lipids. I ran on with that one once again. Diffusion of ions through channels is generally slower than free diffusion through the lipid bilayer because channels occupy a small fraction of the membrane's total surface area than lipids. Still, facilitated diffusion through channels is a very fast process. More than 1 million potassium ions can flow through a potassium channel in 1 second. Wow. A channel is said to be gated when part of the channel protein acts as a plug or a gate. Changing shape is one way to open the pore and another way to close it. Some gated channels randomly alternate between the open and closed position. Others are regulated by chemical or electrical charges. Others are regulated by chemical or electrical changes inside and outside the cell. When the gates of a channel are opened, ions diffuse into or out of the cell, down their electrochemical gradient. The plasma membranes of different types of cells may have different numbers of ion channels and thus display different permeabilities to various ions. Carrier-mediated facilitated diffusion. In carrier-mediated facilitated diffusion, a carrier, also called a transporter, is used to move a solute down its concentration gradient across the plasma membrane. The solute binds to a specific carrier on one side of the membrane and is released on the other side after the carrier undergoes a change in shape. The solute binds more often to the carrier on the other side of the membrane with a higher concentration of solute. Once the concentration is the same on both sides of the membrane, solute molecules bind to the carrier of the systolic side and move to the extracellular fluid as rapidly as they bind to the carrier on the extracellular side and move into the systole. I'm going to do that again. Once the concentration is the same on both sides of the membrane, solute molecules bind to the carrier on the systolic side and move out to the extracellular fluid as rapidly as they bind to the carrier on the extracellular side and move into the systole. The rate of carrier-mediated facilitated diffusion is determined by the steepness of the concentration gradient across the membrane. The number of carriers available in a plasma membrane places an upper limit called the transport maximum, or the rate at which facilitated diffusion can occur. Once all of the carriers are occupied, the transport maximum is reached, and a further increase in the concentration gradient does not increase the rate of facilitated diffusion. Thus, much like a completely saturated sponge can absorb no more water, the processes of carrier-mediated facilitated diffusion exhibit saturation. Channel-mediated facilitated diffusion of potassium ions through a gated potassium channel. A gated channel is one in which a portion of the channel protein acts as a gate to open or close the channel's pore to the passage of ions. Substances that move across the plasma membrane by carrier-mediated facilitated diffusion include glycose, fructose, galactose, and some vitamins. Glucose enters many body cells by carrier-mediated facilitated diffusion as follows. Glucose enters many body cells by carrier-mediated facilitated diffusion as follows. To repeat. Glucose binds to a specific type of carrier protein called the glucose transporter, GLU, capital T, on the outside surface of the membrane. As the transporter undergoes a change in shape, glucose passes through the membrane. The transporter releases glucose on the other side of the membrane. The selective permeability of the plasma membrane is often regulated to achieve homeostasis. For instance, the hormone insulin, via the action of the insulin receptor, promotes the insertion of many copies of glucose carriers into the plasma membrane of certain cells. Thus, the effect of insulin is to elevate the transport maximum for facilitated diffusion of glucose into the cells. With more glucose carriers available, body cells can pick up glucose from the blood more rapidly. An inability to produce or utilize insulin is called diabetes mellitus. That's in Chapter 18. Osmosis. Osmosis is a type of diffusion in which there is net movement of a solvent through a selectively permeable membrane. Like the other types of diffusion, osmosis is a passive process. In living systems, the solvent is water, which moves by osmosis across plasma membranes from an area of higher water concentration to an area of lower water concentration. Another way to understand this idea is to consider the solute concentration. In osmosis, water moves through a selectively permeable membrane from an area of lower concentration to an area of higher concentration. During osmosis, water molecules pass through a plasma membrane in two ways: by moving through the lipid bilayer via simple diffusion as previously described, and by moving through aquaporins, integral membrane proteins that function as water channels. Osmosis occurs only when a membrane is permeable to water, but not permeable to certain solutes. A simple experiment can demonstrate osmosis. Consider a U-shaped tube in which a selectively permeable membrane separates the left and right arms of the tube. A volume of pure water is poured into the left arm, and the same volume of a solution containing a solute that cannot pass through the membrane is poured into the right arm. Because the water concentration is higher on the left and lower on the right, net movement of water molecules, osmosis, occurs from left to right, so that the water is moving down its concentration gradient. At the same time, the membrane prevents diffusion of the solute from right arm into the left arm. As a result, the volume of water in the left arm decreases and the volume of solution in the right arm increases. You might think that osmosis would continue until no water remained on the left side, but this is not what happens. In this experiment, the higher the column of solution in the right arm becomes, the more pressure it exerts on its side of the membrane. Pressure exerts in this way by a liquid, known as hydrostatic pressure, forces water molecules to move back into the left arm. I'm going to repeat that. Pressure exerted in this way by a liquid, known as hydrostatic pressure, forces water molecules to move back into the left arm. Equilibrium is reached when just as many water molecules move from right to left during the hydrostatic pressure as move from left to right due to osmosis. To further complicate matters, the solution with impermeable solute exert a force called the osmotic pressure. Again, to further complicate matters, the solution with impermeable solute also exerts a force called the osmotic pressure. The osmotic pressure of a solution is proportional to the concentration of the solute particles that cannot cross the membrane. The higher the solute concentration, the higher the solute's osmotic pressure. Consider what would happen if a piston were used to apply more pressure to the fluid in the right arm of the tube. With enough pressure, the volume of fluid in each arm could be restored to the starting volume, and the concentration of the solute in the right arm would be the same as it was at the beginning of the experiment. The amount of pressure needed to restore the starting condition equals the osmotic pressure. So, in our experiment, osmotic pressure is the pressure needed to stop the movement of water from left tube into the right tube. Notice that the osmotic pressure of a solution does not produce the movement of water during osmosis. Rather, it is the pressure that would prevent such water movement. Normally, the osmotic pressure of the systole is the same as the osmotic pressure of the interstitial fluid outside the cell. Because the osmotic pressure on both sides of the plasma membrane, which is selectively permeable, is the same, cell volume remains relatively constant. When body cells are placed in a solution having a different osmotic pressure than systole, however, the shape and volume of the cells change. As water moves by osmosis into or out of the cell, the volume increases or decreases. A solution's tonicity, tonic meaning tension, a solute's tonicity is a measure of the solution's ability to change the volume of cells by altering their water content. Any solution in which a cell, for example a red blood cell, maintains its normal shape and volume is an isotonic solution. The concentration of solutes that cannot cross the plasma membrane are the same on both sides of the membranes in this solution. For instance, a 0.9% NaCl solution, which is 0.9 grams of sodium chloride and 100 milliliters of solution, called the normal physiological saline solution, is isotonic for red blood cells. The red blood cell plasma membrane permits the water to move back and forth, but it behaves as though it is impermeable to salt and chloride. The solutes, any Na or Cl ions that enter the cell through channels or transports are immediately moved back out by active transport or by other means. When red blood cells are bathed in 0.9% NaCl, water molecules enter and exit at the same rate, allowing the red blood cells to keep their normal shape and volume. A different situation results if red blood cells are placed in the hypertonic solution, a solution that has lower concentrations of solutes than the systole inside the red blood cells. In this case, water molecules enter the cells faster than they leave, causing the red blood cells to swell, eventually to burst. The rupture of red blood cells in this manner is called homeolysis.